Hello everybody, we're the Brown team. This is our first year doing iGEM. So yeah, who are we? We are zany people, but we are zany people who work very hard and are very pleased with the research we're doing. This year we were able to have a team of 9 undergraduate students. We had 4 graduate students advising us and faculty from across the campus coming in when we needed help in different areas. Starting out, the idea sort of evaporated at the beginning, sort of condensed at the beginning of second semester this past year. And well, it's sort of same story we heard from the Purdue team. It's sort of a lot of interest among undergraduates, but no funding, no lab space, and no real idea of what we were doing. So we started out by forming a journal club, and we read a whole bunch of the seminal papers in the field. Gathered funding from every department we had access to, which amounted to a great deal of begging, but worked out pretty well. For the course of the summer, we were able to fund 8 students, 2 full-time and 6 part-time. Let's see. Oh, yes, and we do say the different concentrations we had. So something that was an emphasis when we were looking for who could be on the team was variety in backgrounds, because it's very definitely an interdisciplinary science, and I feel that's brought a lot to what we studied. We've had a lot of sort of crazy ideas, some of which we threw out quickly, some of which we threw out gradually. We had this original idea, which was to build a circuit that would allow cells to count their generations. We got in contact with another lab that was working on this. They said, well, we've got a lot more facilities than you have, and we've been working on it for 2 years, so maybe for your first project you want to do something else. So we threw that out. But we've wound up with a couple of projects that we've had a fair amount of success on. So without going on too long, I think I hand it off to you.
So I'm going to talk to you guys about the first project that we did, which was, okay, great. Which was the project of Bacterial Freeze Tag. So I don't know if you guys know what freeze tag is, but it's a very popular childhood game. And in bacteria, it would work something like this. So you would have one cell which is able to move, like this guy. And then you'd have a cell that was the it cell, and that cell would release a signaling molecule. The cell would now stop and turn blue, because blue means cold. And then another cell would release another signaling molecule, and the cell would now turn yellow, and it would be able to regain its movement. So how do we do this? Now, we based this on the idea that bacteria require a certain motility protein called MOTB to move. The Penn State team used this same protein. And this is just a flagellar protein that allows movement of the flagella. And without it, bacteria cannot move. So we used MOTB knockouts for our bacteria. So first of all, we have the it cell. And I'm going to go through all this in a second. And then we have the receiver cell. Now, the receiver cell has 3 functions. 1, it has to receive the AHL signal. 2, it has to actually cause the motility to stop. And 3, it has to have the ability to unfreeze itself if another motile bacteria comes its way and gives us another signal to regain its movement. So first I'm going to talk about the AHL receiver. So when the sender cell, as you saw before, releases its AHL, the AHL will come into the cell, and from a constitutive promoter, TET-R, you get this transcription factor, LuxR. Now, LuxR will bind to AHL. When it binds to AHL, LuxR becomes activated, and the activated form of LuxR can then bind to pLuxR and cause LAC-I expression. Now, this is important. All you have to take out of this, so when there's AHL coming in, you get LAC-I out, okay? The next part of our cell is the freeze machine, as we called it. And this is what would actually reduce the motility of the cell, cause it to stop moving. So let's take a look at that in more detail. So here you have your moving cell. And here's the system. Right now, you have no inhibition from p-LAC-I, because there's no LAC-I in the system. And you are creating MOTB, which is the motility protein, so therefore the bacteria is moving. And you're also creating CI, which inhibits this whole set of genes. And I'll tell you what LAS-I is in a second. But as we said before, if AHL comes into the system, you do get lac-I. This lac-I, in turn, shuts off these 3 proteins because it inhibits p-lac-I. And what that does is it freezes the cell. As you can see, it stopped moving. And that also is going to degrade the CI that was being produced right here. And now PCI is activated and produces more lac-I, creating a positive feedback loop. And you can see this with this schematic, I guess. And lac-I now builds up over here, and the cell becomes completely frozen. All these proteins are now completely shut down. And finally, the cell lights up blue because it's frozen. Okay, so next I'm going to talk about the basis for unfreezing. So there's 3 other proteins that you see here. This LAS-I, LAS-R, and AIIA. So what are these proteins? So LAS-I, what that does is it creates another signaling molecule, AI1, autoinducer 1. Now, this signaling molecule works the same way as AHL. It can diffuse out of the cell, and it will be used for unfreezing the cell. But now notice that this cell only makes AI1 when these genes are being transcribed, when it's in a motile state. But when it is frozen, when lac-I comes in and the cell becomes frozen, these are the 2 molecules that get made. You get LAS-R, which is a transcription factor that binds AI1, and AIIA. Now AIIA, all it does is it just simply degrades any AHL in the system. But the important thing to take out of this slide is the AI1 is being produced only when the cell is in the motile state, and the LAS-R is only being produced when the cell cannot move. So the LAS-R is just waiting for a motile cell to come along, give it AI1, and activate it. And you'll see that in the next slide. So this is the point of Dr. Unfreezer, which will actually cause the cell to regain its movement. So as we said, when the cell is frozen, it's creating this LAS-R. Now AI1 comes in, binds to LAS-R, activates LAS-R, and this activated LAS-R goes and activates transcription from p-LAS-R, creating YFP, turning the cell yellow, and creating CI. Now this is the key, because this is going to short circuit our positive feedback loop. So again, the cell is frozen. There's no CI being produced. But all of a sudden now, we start getting CI, as you saw in the last slide. So this CI comes in, and now lac-I stops being produced. So slowly, lac-I will degrade, as seen here. And when that degradation is finished, that inhibition gets stopped, and these cells are all activated again, and you start getting movement from the cell again. And you get further CI, so again, you get a positive feedback loop that pushes the cell to this state. That's it. And essentially, you may be asking what we did with this actual project. It's a nice plan. We had a lot of issues with cloning. As you can see, the constructs are quite large. But we made preliminary steps along the way. And one of our biggest issues is being able to secrete the AHL in a large enough concentration so that the receiver cell can actually feel its effect and activate the downward steps in the system. Trust him. Oh, sorry. Oh, yeah. Sorry. Oh, just hit the red button.
Okay. Hi, my name's Annie, and I'm going to present on the second project that our team worked on this summer. It's the Tristable toggle switch. And we actually came up with this project based on the bistable toggle switch that Gardner, Cantor, and Collins worked on and wrote about in 2000. And they basically created this system that contained 2 repressible promoters that were constructed in a way that makes them mutually inhibitory. So when one part of the construct is active, the second one is being inhibited. And so we thought, well, they made this work with 2 promoters. What would happen if we used 3? So this is a diagram of what the Tristable toggle switch would look like, but an easier way to look at it might be to break it down into its 3 component parts. And so as you can see, we have 3 constructs, and each one has its own, and each part has its own constitutively active promoter. There's PBAD and PLAC and PTET, but each of these promoters also has its own inducer and inhibitor. And so how this system works is that when we add an inducer, it will bind to the specific repressor and inactivate that repressor and allow the activation of that system. So hypothetically, if we were to activate the PBAD ARIC construct, polymerase would be able to bind, and TET-R and LAC-I would be coded for. Now these two proteins would then inhibit the action of the PLAC and PTET constructs, and so we would get RFP expression but no CFP or YFP. However, we can induce the system by adding IPTG, which now binds to LAC-I and inactivates this repressor, and now the PLAC construct is active. Polymerase will be able to bind, ARIC will be produced, and TET-R will be produced, and CFP formation. And so in this way, we can switch between three, hypothetically, we would be able to switch between three stable states. And so the majority of the time we spent on this project was in creating these three constructs. And although we were able to finish the PBAD construct, but due to time constraints, we weren't able to finish all of them. But modeling was another important aspect of this project. And so LK here will give you more information on that.
Okay. So I'm going to talk a little about the modeling. And we worked on the modeling in tandem while we were constructing the Tristable toggle switch. So we created a deterministic model based on work done in these two papers, again from the Collins lab. And the reason why we wanted to go ahead and model this system was because we weren't sure about the stability of the system, whether if we flick the switch, whether it'll stay in one particular state. So we were worried about that because there's leaky expression of the genes even under inhibition. So we wanted to try to model that. So how do I? Okay, so onto the model derivation. So the end goal is at the bottom, we want to be able to get some sort of differential equation for the number of protein monomers. And with this equation, we want to solve it numerically to see how it changes in time. So we want to look at the reactions that lead to the production of these proteins. So we divide them into two categories, the fast reactions and the slow reactions. The fast reactions are what we like to call the merizations, the dimerizations, the trimerizations, and even the tetramerizations of the inhibitor, and their subsequent binding with the operon. These are the fast reactions because their time scale is on the order of seconds. So we consider them to be in equilibrium. On the other hand, we have the slow reactions, which is transcription, translation, and degradation. These occur more slowly on the order of minutes. And so we'll write a differential rate equation for these processes. I guess I have to step over here. Okay, so the equations themselves. So right here we have the fast equations, the polymerization reactions. So we're going to look specifically at the LAC-I inhibitor and the polymerization of the LAC-I inhibitor. So it actually forms a tetramer. And so in the first equation, we have two LAC-I monomers forming a dimer, and so on and so forth to form a trimer and a tetramer. And these have an associated rate constant, which since this is a model that we weren't able to test with experiment, we pulled existing values from the literature. And these are fast, so they're in equilibrium. And there's also the binding of the tetramer to the operator at the bottom. And now on to the slow reactions. So the slow reactions are what we'll be able to write a rate equation for. And there are two main equations, which is the expression of the gene when it's being inhibited, which is right here, the bound state, and when it's unbound, this equation. So from the, after I'm omitting a lot of the math, we were able to write an equation this equation down at the bottom. So how do we understand this equation? So right here we have dz dt, which is the rate of production of a LAC-I monomer. And we have two terms right here. There's this first term and this second term. So the first term is the TEDR produced by the PBAD promoter. And so there are 4 different, what are they? Operons, or operator binding sites, sorry. And the second contribution will be from the TEDR promoter. So now, now that you have a working knowledge of these equations, or a basic knowledge, I'll let Jason talk about some of the actual runs that we did with this model.
Okay. So on to the results. So first we ran the model on the inducer list system. And it turns out that the system is stable without any inducer. And this was somewhat predicted because the promoters do have different strengths. So it turns out PLAC appears to be a stronger promoter than both the pTEDR and pBAD. So in the absence of inducer, we had a steady state. Then we tried the model. We ran the model in the presence of the different inducers. I've pictured here the model in the presence of tetracycline. And as we expected, the pTEDR promoter is dominating, and we are getting expression of YFP. And so our model confirms our hypothesis in the presence of the different inducers. The other inducers yielded similar results. So now the true test, which is the test for stability. Will the system remain stable in the absence of any inducer? And this was where we thought we'd have problems with leakiness from the different promoters. So we figured out a pretty simple scheme to test this, which was simply to plug in the final protein concentrations, similar to those in the previous slide, and to test whether the state would remain stable. And ta-da, it does not. So, in light of this somewhat disappointing result, it's still a preliminary model. And again, like we said, we used only parameters based on those in the literature. So we are, in the future, we're planning to experimentally obtain more appropriate and more realistic values for the model. And hopefully these will yield better results. And also to run parameter scans, which will hopefully yield regulatory elements that are capable of creating tri-stability. 2 minutes. Okay. And finally, we hope to add stochastic variation into the model, which we didn't do with our model right now because it didn't work even without stochastic variation. So there we go. Okay, so conclusions. Like we said before, we worked on these two projects, the Tristable toggle switch and freeze tag. And although our model predicts failure at the moment, we're going to strive on and work more on the model, and also work more on the construct. And hopefully we'll be able to obtain some more reliable parameters. And secondly, we're going to keep working on the freeze tag machine and finish up with the sender and receiver parts, which could be useful for other projects as well. Yeah. And finally, I'd like to thank Gary Wessel, who is the faculty advisor for our project, and also the other various faculty members involved, and also Ruth Simmons, president of Brown, who was very generous in funding the team. Thank you. And there's an extensive derivation of our model on the wiki, and also the code if you'd like to take a look. Sure.
Okay, so you said your model predicts that the construct that you would make would not be stable. I mean, I guess that it would converge on some intermediate level of expression of all of the things?
Actually, it simply returns to the natural steady state in the absence of inducer. Because as you can see right here, we have the, originally, with tetracycline, we have expression of, we have expression of YFP at the top from the TEDR promoter. Then when we switch, or when we remove the tetracycline, it switches to expression from the PLAC promoter, which is the same as the natural steady state. So we also tested the model with essentially identical promoters, simply changing the names, but saying they all have the same reaction rates and so forth. And it did remain stable. So that's another thing to look into. It seems that if we have a system that has a more intermediate steady state in the absence of inducer, it's more likely to lead to tristability, which makes sense. We also tried making combinations of bistable switches from the different components of the system, and they were all stable, so it seemed to verify the model to some degree.
Yeah, I was just wondering, did you model cooperativity in your tristable switch model?
What is that?
Cooperativity.
Cooperativity?
Cooperativity.
In what sense to me?
As in, did you take into account, if say one site on promoter is filled, is more likely for another one to be filled next to it? Because that's one of the conditions for a bistable switch. You've got this cooperativity involved. That could be weird going forward in the model. I don't know.
Yes, I believe that was taken into account with the different reaction rates and whatnot within the model.
Any additional questions? All right, let's thank our speakers.